Well, I'm very pleased to introduce Dr. Cruchaga. We have not met in person until just today. Very interested to hear about his work. He is a human genomicist with expertise in multi-omics, informatics, and neurodegeneration. He completed his PhD in biochemistry and molecular biology at the University of Navarra in Spain. And in his first postdoc, he began focusing on Alzheimer's disease and Parkinson's disease. He came to Wash U to finish his training in the lab of Dr. Alison Goat. And in 2011, he started his own lab studying the genetic architecture of neurodegenerative diseases. In 2019, he became the scientific advisor of the McDonald Genome Institute at WashU and founding director of the Neurogenomics and Informatics Center. He has a long list of accomplishments, but his interests remain focused on using human genomic data to identify and understand the biology of neurodegenerative disease. In sum, his mission is to increase understanding of the molecular underpinnings of neurodegenerative disease to improve human health. Thank you for sharing your work with us today, Dr. Cruchaga, and we'll turn over the format to you.
Yeah, thanks very much. I'm trying to share my screen, but it looks like someone needs to allow me to share the screen. So, okay. So in any case, I'm very happy to be here today to present our work. As you have heard, my research is focused on using human data, human genetic and omic data to understand the biology of complex traits, mainly neurodegenerative traits, and to try to create prediction models and identify new interventions. So today I have a small introduction about Alzheimer's disease. Obviously is the most common neurodegenerative disease, but also I will present two projects that I think is a good example of how we can use all of these human genetic and omic data to understand the biology of Alzheimer's, but also to identify genes that are involved in healthy aging. So let's start. If you have any questions, you can interrupt me, and I also have been told that you can ask questions on the chat and we will go through those later. So let's go through it. So fortunately, I imagine that most of you, if not all, are very familiar with Alzheimer's disease. Alzheimer's disease is clinically characterized by gradual onset and progression of memory impairment, and also deficit in executive functioning and language. From a neuropathological point of view, we know that Alzheimer's disease is characterized by the presence of extracellular plaques, which are compounds for A beta peptide, and intracellular deposits of a protein called tau that form tangles. So these are the main hallmarks of Alzheimer's disease. We also know now that there is a lot of inflammation involved in Alzheimer's disease, and also there are other proteins that can be found in Alzheimer's disease brains, including TDP-43. As I mentioned, Alzheimer's disease is the most common neurodegenerative disease, and there are more than 5 million people in the US with Alzheimer's disease at this moment, and around 100,000 deaths per year are attributed to death. The major risk for Alzheimer's disease, as you can see here, is age, and you can see that the percentage of individuals with disease increased significantly from 65 to 85 years old. And obviously this is going to be a problem in some developed countries in which the mean age is highly increasing. So by 2025 or 27, it's expected that will be more than 25 million people with Alzheimer's in the US. So clearly this is a big issue, and obviously it's opposite to healthy aging. So we need to find a way to solve this. From a genetic point of view, there are two major types of Alzheimer's disease. One is the sporadic form of the disease, which is the most common form and is associated with the late age at onset. It's considered to be a complex trait with genetic and environmental factors to be associated. The other form is the familial or inherited form, which is more rare and is associated with earlier onset, and normally is autosomal dominant. There is a very good study here in Washington University focusing in this familial form, the Dayan study, which is an international study focused on families with autosomal dominant AD. Most of the talk today will be in sporadic, which is the most common form. This is a similar slide to before. The early onset or familial is very rare. The symptoms appear before 65 years old. There are individuals in the Dayan study that have an onset in the 35, so it's very early onset. And we know that mutations in 3 genes, APP, preselin-1 and preselin-2, are causing this disease. So this is one single mutation causes the disease. It's early onset and is 100% penetrant. On the other side, late onset AD is the most common form of the disease, as I mentioned, between 90 to 99% of the total AD cases. Normally, the age at onset is higher than 65, and as I mentioned, it's a complex trait. Late onset AD also is called sporadic, but even it's called sporadic, 40% of the cases have one or more affected first-degree relative. So obviously, family history is also one of this, the second stronger risk factors. From a genetic point of view, the most important risk factor for sporadic AD is ApoE. It has an O ratio between 2 to 8, meaning that having ApoE4 increases the risk to develop AD about 8-fold. So it's quite dramatic. But more than 50% of the AD cases doesn't carry an ApoE4 allele. So we know there are more things going on. And I will go through that later and why that is important. In any case, from a genetic point of view, from the early onset familial AD, as I mentioned, we know that 3 genes are causing disease. And the first gene was identified in 1991 by Alison Goy, who was here. And it was a classic family-based genetic study. And this is the family in which the mutation in APP was found. And this was done by classic linkage analysis. After that, mutation in 2 other genes, preselin-1 and preselin-2, were identified. This is important because the mutation of these 3 genes are all part of the same pathway. So APP can be processed. It's a membrane protein that can be processed, leading to some soluble forms. And there are 2 potential pathways, the no-amyloidic pathway and the amyloidotic pathway. And that depends how this APP is processed. If it's processed first by alpha secretase, we have this APP alpha, which is soluble, and really nothing happens. If, on the other hand, if APP is first cleared by beta secretase, then we have two forms. That is a soluble APP beta and another APP which fragment, which is again processed by the gamma secretase, which preselin-1 and preselin-2 are gamma secretase, leading to this product, which is the A beta peptide, which aggregates and form these plaques. It is important that A beta aggregates are present in all the forms of AD, whether they are familiar or sporadic. The most interesting thing is that all the pathogenic mutations are in APP that are associated with this alpha, beta, or gamma secretase cutoff, or in APP in these specific positions. So all these mutations are part of the same pathway. So clearly A beta aggregation is important for Alzheimer's disease. In any case, this is for familial AD. What do we know about the genetics for sporadic Alzheimer's disease? The genetics for sporadic Alzheimer's disease is a little more complex, and we need a much larger sample size in order to identify genetic variants. This is one of the latest genetic studies for Alzheimer's disease. It was published in 2019, and almost 90,000 individuals were included in this analysis. So you can see we really need a large collaborative consortium to achieve these studies. This is the summary of the study, and this is what is called a Manhattan plot, because theoretically these signals looks like the profile of Manhattan. And what we are seeing here is a summary of all the genetic variants that were tested. Each dot represents a genetic variant, a single nucleotide polymorphisms, and they are sorted by chromosome and base pair position. So here we have the 23 chromosomes that are in the human genome. In the y-axis scales, we have the p-value in a log 10 scale. So in these studies, because we analyze millions of SNPs, in order to be considered significant, the p-value needs to, the SNP need to have an association higher than 10 to minus 8, which is this line you can see here. So only those SNPs that are above this line are considered significant. As you can see here, there are many signals that pass that threshold, which is good because now we identify more than 20 or 30 genetic regions associated with AD risk. As you can see here, we have ApoE that I already reported, but we also have many other genes. I want to highlight two of them because I want to talk about them later. One is called TREM2, some of you may already know, and the other is the MS4A gene cluster. Okay, so as you can see here, these studies has been very successful in identifying these genetic variants associated with AD risk, but they have two main problems. And this is important to say very clear, and I will explain how we can use the omic data to try to address those problems. The first problem is that the effect size of these signals is very small. So I told you that the effect size of ApoE, the odds ratio is between 2 to 4 or 8, depending on the study. You may see here that BIN1 is similar to ApoE, but this is a problem with the plot because the association of ApoE was so high that they need to cut this plot. The association with ApoE was around minus 500, and here we're in the minus 40 range. So if we compare the effect size of ApoE with other regions, is much different, right? The odds ratio for ApoE is around 4.4, which is a risk factor. ApoE2 is a protective factor, odds ratio about 0.5. Everything else is between 0.9 and 1.1. So the effect size is very small. And some of you may ask why we are doing this very large, very expensive study to identify genetic variants that they don't have even a small effect size. And this is a valid question. The question is now that we have so many of these hits that when we aggregate all of them together, even without knowing anything about the biology, we can create very good prediction models. And this is a study published also a few years ago that they put together the 20 signals that were significant together with ApoE, A and sex, and they were able to have a prediction model with an area under the cube around 0.79, which is pretty good. And this is close to be significantly or clinically relevant. And this is already a paper that was around 5 years ago. This paper has not been rerun with the new hits, that is almost a double, but we are going to get better and better. It is important that this is baseline risk, meaning that this is the risk with one individual has since they are born, because these are germline variants, right? So we can really predict from the beginning of life where the risks of someone develop AD later. So I think that's quite amazing. So even the effect size is low, this can be very useful to create prediction model. The second problem of these studies is that even in that Manhattan plot, there is a gene name in top of each region. Sometimes, or most of the times, we don't know what is the functional gene driving the association. This is a locus plot, so it's basically a zoom on one of these regions. And in this case, you can see that the SNPs associated with AD risk are around one single gene. This is a SOROR1. We know that SOROR1 is also involved in A-beta processing, and there are some functional studies that confirm that this gene is relevant to Alzheimer's disease. So in this case, we know that the functional gene is SOROR1. But in other cases, when we have a gene name, we really don't know the gene because there are many genes that are covered by this signal. And we still don't know the biology of every gene in the genome. So we don't know which genes are involved in Alzheimer's disease. This is one example. And this is the other example in the MS4A region, which I mentioned. And you can see there are many MS4A genes, A3, A2, 6A, 6A, 4A, and so on. So how we go from a p-value to biology? That's the second problem. So as I mentioned, GWAS has been very successful in identifying statistical significant variants associated with complex traits, but have failed to really understand the biology of these traits. We know that some of these signals explain a very small proportion of the phenotype, so whether this is relevant or not, right? So the question is how we can leverage other omic data to go beyond these simple regions and p-values and really understand the biology of Alzheimer's and healthy aging in general. And that's what I'm going to try to do today. So the goal of my lab is to understand the phenotypic variability, to understand why some people develop Alzheimer's disease, why some people are resilient and they really don't develop Alzheimer's disease, why the age at onset for some people is earlier than others. And we think that by doing this, we are going to identify novel genes and pathways, novel molecular biomarkers, create better prediction models, and identify drug targets. Most of the studies so far has been focused on genomics alone, only in DNA. One of the reasons is because the technology to analyze DNA is very good, but that is not enough. If we want to really understand this phenotypic variability, we need to go beyond that first layer. And we need to study the epigenome, the transcriptome, the proteome, the metabolome, and the lipidome as well. And we have the technology these days to do that. And that's what we have been trying to focus in my lab. We are trying to molecularly phenotype clinical cohorts, generating multiple layers of omic data. So this is exactly what we are doing, and this is a very brief study design of what is going on. We are not only focusing we are focusing on brain tissue, on CSF, so spinal fluid, on plasma. We are using also iPSC models. And for all of these tissues, we are generating genetics, proteomics, epigenetics, metabolomics, transcriptomics, and lipidomics, and so on. And we are doing this from sporadic AD, Mendelian AD, also cognitive normal individuals. And the goal, as I mentioned, is to identify these protected variants, create prediction models, and identify drug targets. So let me explain you how we can use the data to understand a little about the biology of Alzheimer's disease. And we are going to focus on one of those genes that I explained before, which is MS4A4A. MS4A4A was reported, I think, in 2013. And there are many genes in that region. So nobody has done anything about those genes because we don't know the biology of those genes. We don't know which cell types are expressed. We don't know what is the mechanism modifying AD risk. So let's see how we can put all of that together using human and multi-omic data. So We are going to switch gears a little and then everything will come back. In a few years ago, 2 different papers reported the association of TREM2 with Alzheimer's disease. This was a very important finding for 2 different reasons. The first reason is because in this case, we identify a gene and not a region. We identify a gene because in this case, we identify functional variants in TREM2. And the second important thing is that TREM2 is clearly not a neuronal gene. It's not associated with ABER and tau. TREM2 is associated with microglia, activity, immune response, and inflammation. All of these led to a shift in the field, realizing that microglia and inflammation was very important for Alzheimer's disease and aging in general. And until that, the field was very neuronal centric. So all of this switched quite a lot about why microglia is so important for Alzheimer's disease. Also, the effect size of these TREM2 variants was very, very high. It was as high as ApoE. So that was the 3 things make this finding very important. Very high effect size, we knew the gene, and it was a new pathway. So based on that, I started collaborating with Laura Picchio, and we decided to measure soluble TREM2 in CSF. And in our first paper that we published in Acta Neuropath, we found that CSF TREM2 was increased in early stages of AD and correlate with CSF tau levels. There were several variants in TREM2. There's different type of variants, and we found that those different variants has also different soluble TREM2 levels. One of the variants was associated with higher level, and other variants was associated with lower levels. So that makes study TREM2 a little complicated, but also suggests that there were different pathogenic mechanisms for TREM2. In any case, the most important part of this paper was that CSF TREM2 levels was potentially a informative biomarker for AD. And what we decided to do that was to perform a genetic screening of CSF TREM2 levels. And we performed what is called a GWAS, so similar to what I saw before for AD risk. And this was a relatively small study. The study I showed you for AD risk include 90,000 individuals. This include 800 individuals. But it was a very successful, and we are very happy that we ran this analysis. It was a high risk, high reward. And that's what we found. Only with 800 individuals, we found a very, very strong signal in chromosome 11. And this is not any region randomly. It's exactly the same region as the one for Alzheimer's disease risk in the MS4A gene cluster. It's exactly the same variant. You know, we were analyzing 8 million variants. What are the chances that we found the same variant associated with AD risk? So this is a very, very important finding, because only with this, we are now start putting a context, a biological context of the association of MS4A with Alzheimer's disease. What we are saying here is that there are genetic variants in MS4A that are modifying soluble TREM2 levels. And we think this is the mechanism of action. MS4A4A modified risks for AD by modifying TREM2 biology. And this is very, very big because we didn't know anything about MS4A before. So now we know that what are the mechanisms. We know that TREM2 is a microglia gene. And obviously now we realize that MS4A is also a microglia gene. So I think this is very important finding that we are now putting a biological context. Another important finding is that we found 2 independent signals in this region. This is the original signal, and you have to believe me, it's exactly the same signal as before. But the important thing is that we identify another signal, and in this case, this is a coding variant in MS4A4A. So all of these genes that we have in the region, now we have a candidate gene that we can start modeling and targeting. Another important thing that we need to mention is that this is a protective variant, meaning that this variant is associated with lower AD risk and also associated with higher soluble TREM2 levels. When TREM2 was identified, because it was associated with immune response and inflammation, it was not clear whether we need to activate microglia, increase TREM2 or decrease TREM2 levels in order to have a positive benefit. And this study clearly indicates that we need to increase soluble TREM2 levels in order to decrease Alzheimer's disease risk. This is just a forward plot of these two variants. And basically what we did was measure the same variants in multiple studies, and we were able to replicate the association of these studies in other cohorts of sporadic AD, but also in autosomal dominant AD. And here we have Diane, that we will replicate the protective variant effect, but also the coding variant. So this was very important. As I mentioned, another question we asked in this study is whether TREM2 is involved in AD in general or not, because TREM2 was initially reported to be associated with AD because there are some very rare variants in TREM2 that show that association, but we didn't know if TREM2 in general was. So we performed what is called a Mendelian randomization approach. This is a statistical method combining the summary statistics from our analysis with TREM2 levels and those with AD risk. There is some statistics behind and mathematics that I'm not going to go behind. The question is, we found a very significant association indicating that TREM2 is involved in AD in general, and not only in those with pathogenic mutations. Another important thing is the slope of this plot, which is negative, indicating again that higher soluble TREM2 levels are protective. And this is very important. We want to increase soluble TREM2 levels. We want to increase the activation of microglia, at least before the clinical onset. So all of this is statistics and p-values and whatnot. How do we know this is real and not just some program error? So in order to functionally validate this, we collaborate with Celeste Katz and Laura Piccio again to validate this using human macrophages. What we did, we used primary human macrophages because it's a good proxy for microglia cell lines. And we know that both TREM2 and MS4A are microglia cells. So what we did was to overexpress using lentivirus, overexpress MS4A or knock down MS4A with shRNAs. And this is quite complicated because it's difficult to transfect macrophages. In any case, we were able to do this, and when we increase the expression of MS4A4A, we also see that there is an increase of soluble TREM2 levels. And when we knock down MS4A4A, we also see that the soluble TREM2 levels decrease. And this is in the same direction that we predicted based on the genetics. And this is very important because what we are demonstrating here is that we can regulate TREM2 levels and therefore AD risk by targeting MS4A4A. We performed this with another two genes, MS4A7 and 6A, and we didn't see a significant effect. At this moment, we cannot discard that those genes are also involved, but we can say that at least MS4A4A is involved or interacting with TREM2. We also performed some colocalization analysis, and we can confirm that MS4A4A and TREM2 colocalize both in the membrane and in the lipid rafts. So we still don't know what is the mechanism by why TREM2 and MS4A interact. We are still working on that. I think that any good studies those that have more questions and the end that those that have an answer, but we have some hypotheses. We know that TREM2 goes to the membrane with this cleavage to form this soluble TREM2 that is the one that we're measuring. And we know that MS4A is an intermembrane protein, and somehow, based on the studies, we think that MS4A affecting this cleavage. So we are now doing additional research in order to determine whether this is real or not, but at least we know that MS4A is the major regulator of TREM2. We identified this in CSF. There was a paper published last week in Nature Genetics that published many other things. And one of the sentences was that they were able to replicate this association in plasma. So this is a real association. In any case, to finalize the part, we can say that MS4A represents the major regulator of soluble TREM2. There are two independent signals in MS4A. The strength of the association is comparable to ApoE. So it's a very large effect size. And that's why we were lucky in the end. And that's the reason we were able to identify this association with only 800 individuals. The most important is that MS4A is also associated with AD risk. So we are putting a biological context of the initial association of MS4A with AD risk. MS4A modifies AD risk by modifying TREM2 biology, and this is very important. We also demonstrate that TREM2 plays a role in AD in general, and higher levels are protective. MS4A co-localize and co-express with TREM2. And we have demonstrated that we can modify TREM2 levels by targeting MS4A. So this is a new therapeutic target. And I'm not just saying this because it looks good. Now we are collaborating with Alector, which is developing antibodies for MS4A for Alzheimer's disease, and they are measuring TREM2. The idea is to control TREM2 levels, and they are in preclinical trial. So this is a good example of how we can combine genetics and proteomics to understand the biology of the disease and identify therapeutic targets. I don't know if that drug is going to work, but I think it's a good example of how we can move from that Manhattan plot to biology. Okay, so that's one example. I'm going to switch gears now a little about how we can identify protective variants. And we know that Alzheimer's disease is characterized by neuronal loss and gliosis. And there has been out there a lot of studies using Alzheimer's disease brain gene expression to identify genes and networks associated in the disease. Most of these studies has done with bull tissue. And obviously that's a problem because if you have RNA-seq individuals with Alzheimer's and controls, the cell proportion is going to be different, and different cell types express different type of genes. So you can have a very biased approach, right? You can just have genes that looks like are associated with disease, but they are just associated with cell proportion. So in the lab, we were thinking how we can address this, or how we can correct by cell proportion in these assays. And also, can we quantify neurons and determine genetic variants associated with neuronal proportion and aging? And that was the goal of this second part. This is a summary of entire paper, but I want to go through the high-level expression, high-level summary. So what we decided to do was to use this gene expression data to perform a digital deconvolution. So we can basically estimate the proportion of neurons, astrocytes, oligodendrocytes, and microglia just looking at the genes that are expressed and the gene expression. So in order to do this, we create a synthetic panel. So we use RNA-seq that was specific for neurons and these cell types and microglia, or iPSCs, in order to create a reference panel. And after that, we create an algorithm to estimate the proportion of cells in each single experiment. And we were able to validate this in multiple data sets. And we use public data from autosomal dominant AD, including Dayan, the 90DRC here, and public data set from Mount Sinai and Mayo. We were able to demonstrate that this approach correlates very nicely to what's expected. We demonstrate that there is a decrease of neuronal proportion in AD brains, as we expect. We also see that that was more relevant in autosomal dominant AD. And in TREM2, for example, we didn't see that much neuronal loss. In any case, all of that to say is that we are able now to estimate the proportion of neurons, oligodendrocytes, astrocytes, and microglia in any brain RNA-seq data. So what we did was to run that algorithms in a total of almost 1,700 samples from multiple studies. So we download these studies and process everything from scratch and really calculate the proportion of neurons, astrocytes, and so on. And we were very interested in neurons. So in the end, what we have is we have the proportion of neurons in each individual. So this is the individual level. So this is now our new phenotype. And we ask whether there are genetic variants associated with neuronal counts in all these data sets. And we did again a GWAS analysis. And what we found was very relevant. We found a signal that was genome-wide significant, in this case in chromosome C7. And this SNP is not any SNP randomly. It's a SNP is located in TMEN106B. And you can see here the heat, the locus sum. So the important thing is that TMEN106B and these variants are variants that some of you may know because this has been reported to be associated with multiple traits. It has been shown to be associated with AD risk in the latest GWAS for Alzheimer's disease with 1 million people. Initially, it was reported to be associated with frontotemporal dementia, specifically individuals with granulin levels. And we also demonstrate that TMEN106B regulate granulin levels. Both granulin and TMEN106B are lysosomal genes. TMEN106B has been recently reported to be implicated in COVID as well. So what's going on with this? Why TMEN106B is involved with so many traits? So what we decided to do, because in this study we have individuals with Alzheimer's disease, we also have controls, common mind, have a high age range. So where this association is coming from, this is driven by Alzheimer's disease, this is driven by controls, or what's going on. And the most interesting thing was that we found a strong association with Alzheimer's, as we expected, and other neurodegenerative diseases, including FTD, again, as expected. But the most interesting thing is that we found that this variant was associated with neuronal count in all controls, but not in young individuals. And the cutoff here is 65 years old. So somehow this variant is regulating neuronal survival starting in 65 years old. So what we have here is a general protective factor, because this variant is associated with higher neuronal count. So individuals with this variant has less risk of develop AD or any other neurodegenerative diseases, or even somehow COVID. So just to summarize this part, we start this project because gene expression analysis studies in brain can be confounded due to cell composition. And we have developed a new method to estimate cell proportion and correct for that. The most important thing is that we found that TMEN106B is associated with neuronal proportion, replicates in multiple studies and multiple brain regions, and is a protective factor for aging. It's not an Alzheimer's disease specific, is for aging in general. TMEN106B is a lysosomal protein and is highly expressed in neurons and astrocytes. What is the mechanism by which TMEN106B promote cell survival? That's something we are now trying to study. We are also trying to identify compounds that target TMEN106B, increasing the activity. but that's something we are trying to do. In any case, I think this is a good example of how, two examples of how we can use combined proteomics and genetics to identify potential drug targets, and how we can use transcriptomics and genetics to identify novel protective variants. I want to finalize with a couple more slides about how we are also really using this data. This is an entire paper summarized in one slide, and this is also a good example. If I don't have time today, but someone wants to know more, this is a study in which we combine proteomics and genetics for doing PQTL mapping for going from DAG to drug repositioning. And this is a very good example of, again, combining human multi-omic data to identify drug targets and for drug reposition. And we identify multiple drugs that can be repositioned for Alzheimer's, Parkinson, ALS, and even stroke. In any case, we think that molecular profiling that includes genetics, transcriptomics, proteomics, epigenetics, metabolomics, and any other omics in large and well characterized samples are going to lead to identify new genes and pathways, a deeper understanding of the pathological events. And I hope that the example of MS4A and TIME2 is a good examples to create new prediction models. I have not shown anything about prediction models today. More than happy to talk about that if someone is interested, and novel therapeutic targets, which I think we saw. But we think this human-derived data is the key for a more personalized prediction of disease risks and treatment. And we are really putting a lot of effort in molecularly characterize the samples that we have in the lab. So I just present 2 projects, and there are a lot of people involved in these projects. This is really a teamwork, and I'm just here presenting the work for many people that has been leading this effort. And obviously there is a lot of support from the NIH, from the ADRC, and other foundations. So that's all what I have for today. You have any question or more?
Thank you. Yes, let's thank you very much. I learned a lot. I've never really understood how you approached this topic, and even though I didn't understand a whole lot of things, I did grasp some fundamentals that really helped me appreciate this work a lot more. I'll start out with some questions and hope others will feel that they can jump on and ask as well. You can either put them in the question and answer, or yeah, that'd be the best thing. I, as a gerontologist, as a social gerontologist, always striving to understand more about what age has to do with it, what age has to do with Alzheimer's disease specifically. Yep. And so you did talk a little bit about some protective mechanisms or lack of protective mechanisms with age, but could you articulate for me in my social gerontology terms, sort of why is AD so profoundly attached to somebody's chronological age?
Well, we can start there. I'm going to skip a little with your question. One is the chronological age, another is biological age, right? So that's other thing we can try to understand with genetics is what is the biological age. And, some people develop AD later because biologically they are younger. So in any case, why is an age disease? It's a disease that is very slow. So the pathological events leading to disease starts basically since we are born, but it takes years and years of having some symptoms, right? We know that for Alzheimer's disease, we can identify brain changes 20 to 15 years before clinical onset, but that doesn't mean that the pathological event has started earlier. So this variant MS4A4A that is protective for Alzheimer's disease and increased TIME2 levels, this is since are born. People that has this variant has higher soluble TIME2 levels since the beginning. So they are able to potentially cope with any kind of small brain damage since the beginning, but this is happening since they are born. And same with APOE, right? People are born with APOE, and that leads to more A beta aggregation. So these things are just takes a lot of time to have a presentation, but they are happening even we don't see them.
Thank you. That makes sense. And how could you also throw inflammation into my very basic question of what does inflammation have to do with it? Where does it occur in the process? How is it affecting what you're looking at?
So, I mean, inflammation is a double-sword war, right? Because normally inflammation is always associated with bad outcomes. The question is, in this case, we know that TREM2 is a microglia gene, and microglia is also take care of clean up these debris, especially, you know, if there are some tau protein aggregates. So what we are seeing is that soluble TREM2 obviously is associated with higher microglia activity, and having higher microglia activity seems to be good to avoid to have these protein aggregates in the brain. So if you are, if someone is in the very, very early stages before any clinical symptom, having this activated microglia helps to clean all the debris in the brain. We know that once there are clinical symptoms and there is a lot of neuronal loss, a lot of plaques and tangles, there is also inflammation, and that inflammation is bad. So maybe in that stage, activated microglia is bad because you are creating inflammation in the brain, and this is, you know, leading to more neuronal loss. So it's complicated. But in the beginning, having slightly more activated microglia seems to be protective.
Okay, well, thank you. That helped increase my understanding a bit. Natalie, please jump in if you see any questions or that people would like to ask. I have one more, and then I'll see if somebody else can wants the opportunity. You've been focusing on Alzheimer's disease. I saw that the Michael J. Fox Foundation funds some of your work. Are you doing this? What are you doing? Some of the similar type of studies? What's the overlap with your research with Parkinson's disease?
Yeah, so I mentioned that Alzheimer is characterized by A beta and tau, right? And Parkinson is alpha-synuclein, but we know that 30% of the AD cases have Lewy body dementia, and 30% of the Parkinson has dementia as well. So there is a lot of overlap in these diseases from a clinical, pathological, and genetic point of view. So we are trying to do similar things to Parkinson, but also we are trying to do this with FTD, frontotemporal dementia, and DLB, and so on, because we want to, because in the end, for us, Alzheimer's and Parkinson are also associated with healthy aging. Some of the factors associated with AD associated with healthy aging as well. I'm not doing that much in Parkinson because there is a lot of things ongoing in Alzheimer's, and I have my hands full, but there are other faculties in this new center that they are trying to really do similar approaches with Parkinson. But we are trying to share pipelines, samples, and so on. But I see this is a part of a major phenotype, right, which is aging, healthy aging, and neurodegeneration. I think they all go hand by hand. Obviously, I don't think I can solve all of. I don't think I will be able to solve any of them, but we are trying.
Thank you, Natalie. Any other questions from people?
Doesn't look like it right now.
Okay. Before we finish, are you going to need to put up a slide for continuing ed, or
do that right now?
Okay. Dr. Cruchaga, could you, this is not in your particular presentation at all, but just for the public and for our understanding of where exactly we're at with understanding Alzheimer's, are the new blood tests that are being presented as maybe a solution to some early diagnosis relate to your work?
Yeah, so as I mentioned, we are trying to create prediction models, and that's ongoing. But obviously, the field in general are very interested in creating blood-based biomarkers. At this moment, the best liquid biomarkers are in spinal fluid, and this is how we measure TREM2, right? But CSF is not a great tissue to have access to. So a lot of interest in the blood-based biomarkers. So the best biomarkers in CSF are A-beta, tau, and p-tau. So based on that and based on the biology, there are now new blood-based tests that are measuring A-beta, tau, and p-tau that are very, very good. Before, those tests didn't work because we didn't have methods that were sensitive enough to measure those proteins in plasma. But right now, those methods are very sensitive and they are also very specific. So with a blood-based biomarker now measuring A-beta, it's possible to determine whether an individual has amyloid in the brain. So it's a proxy for having AD pathology independently of the clinical status. Most of the individuals with AD pathology develop AD within 5 years, but there are some people that are resilient, and that's important. But yes, there are now commercial blood-based biomarkers for Alzheimer's disease.
Thank you. One more question has come in from Dr. Burge. Cardiovascular disease seems to be a risk factor for AD. How does this interact with TREM2?
I don't know how to interact with TREM2. I will link it more with ApoE. So ApoE is a stronger risk factors for ApoE, but it's also one of the stronger risk factors for cholesterol levels. So obviously there is a link there. And I would say that is the link is more in the lipid metabolism rather than maybe blood pressure. TREM2 also has some interaction with lipids, even that is still not clear. But I would say for a cardiovascular point of view, ApoE will combine or link both through the cholesterol pathway.
Terrific. Thank you. I think that's all of our questions. I want to thank you for your time. This has been recorded. It'll be on our website for people that want to access it. Thank you. And for people that need that QR code for continuing ed, that was up. Please be in touch with us. We have a lot of good events coming up, and we're certainly off to a great start with Dr. Cruchaga. Thank you very much for your time.
It was a pleasure.
Happy New Year, everyone.